Hey everyone, and welcome. Today, we're diving into the fascinating world of ribosomes. These tiny but mighty structures are essential for all life. Think of them as the miniature protein factories inside your cells. They're constantly working to build the proteins that keep you alive and functioning. So buckle up as we explore the structure, function, and importance of these amazing molecular machines. Ribosomes are complex molecular machines found in all living cells. From bacteria to plants to you, they're everywhere. Their primary job is to synthesize proteins. They do this by reading the genetic code carried by messenger RNA, or mRNA. Essentially, they translate the language of RNA into the language of proteins. Pretty cool, right? Ribosomes aren't just simple blobs. They have a specific structure. They're made up of two subunits, a large subunit and a small subunit. Each subunit is composed of ribosomal RNA, or rRNA, and ribosomal proteins. These components work together to perform protein synthesis. In eukaryotes, the large subunit is the 60S subunit, and the small subunit is the 40S subunit. In prokaryotes, they're the 50S and 30S subunits, respectively. The S stands for Svedberg units, a measure of sedimentation rate during centrifugation. Ribosomes can be found in a few different locations within the cell. Some are free-floating in the cytoplasm, the gel-like substance inside the cell. Others are bound to the endoplasmic reticulum, or ER, creating what's called the rough ER. Ribosomes on the rough ER typically synthesize proteins destined for secretion or for use in the cell membrane. Free ribosomes, on the other hand, usually make proteins that will be used within the cytoplasm itself. So, location matters. Now let's get into the nitty-gritty of how ribosomes actually make proteins. The process is called translation, and it's incredibly precise. First, mRNA binds to the small ribosomal subunit. Then, a special initiator tRNA molecule carrying the amino acid methionine binds to the start codon on the mRNA. Next, the large ribosomal subunit joins the complex, forming a functional ribosome. Now the ribosome is ready to start reading the mRNA code. As the ribosome moves along the mRNA, tRNA molecules bring the correct amino acids to the ribosome. These amino acids are linked together by peptide bonds, forming a growing polypeptide chain. Finally, when the ribosome reaches a stop codon on the mRNA, translation terminates. The polypeptide chain is released, folds into its correct 3D structure, and becomes a functional protein. Transfer RNA, or tRNA, plays a crucial role in protein synthesis. Each tRNA molecule carries a specific amino acid and has an anticodon that matches a codon on the mRNA. Think of tRNA as the delivery service that brings the right building blocks to the ribosome. Without tRNA, the ribosome wouldn't be able to assemble proteins correctly. The genetic code is a set of rules that cells use to translate information encoded within genetic material into proteins. Ribosomes are the key players in this process. Each 3-nucleotide sequence, or codon, on the mRNA corresponds to a specific amino acid. The ribosome reads these codons and adds the corresponding amino acids to the growing polypeptide chain. The genetic code is nearly universal, meaning that the same codons specify the same amino acids in almost all organisms. This is a testament to the fundamental importance of ribosomes and protein synthesis. Ribosomes themselves are complex structures, so how are they made? The process is called ribosome biogenesis, and it's a highly regulated process. In eukaryotes, ribosome biogenesis primarily occurs in the nucleolus, a region within the nucleus. rRNA genes are transcribed, and the resulting rRNA molecules are processed and assembled with ribosomal proteins. The resulting ribosomal subunits are then exported to the cytoplasm, where they can participate in protein synthesis. It's a complex and coordinated process. Because ribosomes are so essential for life, problems with ribosome function can lead to a variety of diseases. Ribosomopathies are a group of genetic disorders caused by defects in ribosome biogenesis or function. These disorders can affect many different tissues and organs, leading to a wide range of symptoms. Some examples include anemia, developmental delays, and increased cancer risk. Understanding the role of ribosomes in disease is crucial for developing new therapies and treatments. Ribosome research is an active and exciting field. Scientists are constantly learning more about the structure, function, and regulation of these amazing molecular machines. One area of focus is developing new drugs that target ribosomes. These drugs could be used to treat bacterial infections or even cancer. Another area of research is understanding how ribosomes evolve and adapt to different environments. The possibilities are endless. So there you have it, a whirlwind tour of the world of ribosomes. These tiny protein factories are essential for all life. They play a crucial role in translating the genetic code into functional proteins. Without ribosomes, life as we know it wouldn't be possible. I hope you've enjoyed learning about these amazing molecular machines. Keep exploring the fascinating world of biology.